Hello everyone, I'm Kajal and I'm a microbiology MSc student. So today I'll give you just a short information about the types of vaccines. So there are several types of vaccines. Each type is designed to teach our immune system how to fight off certain kinds of germs. So and the serious disease which causes, so basically, when scientists create vaccines, what they consider priorly, how our immune system responds to the germ, who needs to be vaccinated against the germ, and what is the best technology or approach to create the vaccines. Exactly, because whatever we are doing, the best part is what's our approach towards that particular thing. So based on a number of these factors, scientists decide which type of vaccine they will make. There are several types of vaccines, as like you can say first is inactivated vaccines, live attenuated vaccines, messenger RNA vaccines, subunit recombinant polysaccharides conjugate vaccines, toxoid vaccines, viral vector vaccines. So these are the types of vaccines which we generally use in the formation of vaccine. So here I'll tell you about one by one of everyone. What is inactivated vaccines? Like in activated vaccines, what we do in this, we use the killed version of the germ that cause a disease. Killed version of that germ means inactivated vaccines usually don't provide immunity. It means don't provide a protection that's as strong as live vaccines give the protection. So many need several doses over time, like booster shots, so in order to get ongoing immunity against diseases. So basically means when means this inactivated vaccines are used to protect against the disease. It's hepatitis A. If you have flu, means shot only. Polio, of course, a shot only, not in a means what we say in not in a mild. Rabies, if you have. So in this type of issues, disease, we use generally inactivated vaccines. So the live attenuated vaccines, vaccines use a weakened means or attenuated form of the germ that causes a disease. So because these vaccines are so similar to the natural infection that they help prevent, so they create a strong, long-lasting immune response. Just 1 to 2 doses of most live vaccines can give you a lifetime of protection against the germ and disease it causes. So, but live vaccines also have some limitations, right? It's not like it is really good, like it is similar to the natural infection that they help to prevent it. So it's like a genius one. So, but live vaccines have some limitations. What are those? That because for example, because they contain a small amount of the weakened live viruses, some people should talk to their health care provide before receiving them. So such as people with weakened immune systems, long term health problems, or people who have had an organ transplant, and they need to be kept cool, so they don't travel well. So that means they can't be used in countries with limited access to refrigerators. So this is the means limitation of this live alternative vaccines. So if I tell you about the live vaccines, means what are the used to protect against this, like the disease which is like measles, if I have mumps, rubella, like that is MMR combined vaccine, it is made up of. So and other is rotavirus, smallpox, you can go with chicken pox, and you can say thing for yellow fever as well. So these are the for the this live attenuated. What are the messenger RNA vaccines which I told you? So messenger RNA is also considered as mRNA vaccines, because research in this researchers have been studying and working with mRNA vaccines for decades, and this technology was used to make some of the COVID-19 vaccines as well. So mRNA vaccines make proteins basically in order to trigger an immune responses. mRNA vaccines have several benefits compared to other types of vaccines, so including shorter manufacturing times, and because they don't contain a live virus, so there is a no risk of causing disease in the person getting vaccinated. So mRNA used protect against basically in this COVID-19 only, which we seen in the COVID section, COVID time pandemic at that time. Another was a subunit, as I told you, the recombinant polysaccharides and conjugate vaccines. What are those? Like subunit recombinant polysaccharides or this conjugate vaccines are specific specific pieces of the germ. Like its protein, it uses a sugar or a capsid which is a causing around the germ. So, because these vaccines use only specific pieces of the germ, they give a strong immune response that the targeted to key parts of the germ. So, they can be used on almost every one who needs them, including people with weakened immune systems and a long-term health problems as well. So, what are it has only one limitations of these vaccines is that we may need to booster shots to get ongoing protection against particular disease. So these vaccines are basically used to protect against Haemophilus influenza type B disease, hepatitis B, or you can say human papilloma virus, which we say HPV, and whooping cough as well. Like it is a part of the DTaP combined vaccine. So pneumococcal disease as in that term as well, and if I say then like meningococcal disease, shingles. So in these, like generally we use this vaccines. What is like toxoid vaccines? Toxoid vaccines are usually use a toxin, means like harmful product which made by the germ that causes a disease. So they create immunity to the parts of the germ that causes a disease instead of the germ itself. So that means of the immune response is targeted to the toxin instead of the whole genome, targeted to the toxin portions only. So like some other types of vaccines, we may need booster shots to get ongoing protection against disease. So toxoid vaccines are generally used to protect against diphtheria, if I have like tetanus. So in this section we use generally these type of vaccines. What is about the viral vector vaccines? Like for decades, scientists studied viral vector vaccines. Some vaccines recently used for Ebola outbreaks. I have used viral vector technology, and a number of studies have focused on viral vector vaccines against other infectious disease, like you can say as Zika, flu, HIV. So scientists use these technologies to make COVID-19 vaccines as well. So viral vector vaccine use a modified version of a different virus as a vector to deliver protection, and several different types of virus have been used as a vector, like including influenza, vesicular stomatitis virus, and measles virus, and adenovirus, which causes a common cold. So adenovirus is one of the viral vector which used in some COVID-19 vaccines being studied in clinical trials. That viral vector vaccines are used to protect against in COVID-19 basically. So these were the things which we haven't seen, and that's all from my side. So what's the future of vaccines? Yeah, we have just learned about that liver. So did you know about the scientists? Those are just still working to create new types of vaccines. Like DNA vaccines are there. So DNA vaccines are easy and it's inexpensive to make. So and they produce strong and long term immunity as a DNA vaccines, and otherwise the recombinant vector vaccines, it is a platform, like if I tell you, it is a platform based vaccines, act like a natural infection. So they are especially good at teaching, good at teaching or introducing the immune system how to fight germs. So basically these all are about these are the types of vaccines from my side. So thank you very much to everyone of you to listen me very carefully. So if you like this information about the vaccine, so kindly just like it, subscribe it, and if you think that it is informative, then kindly share with your friends as well. Thank you so much to everyone of you.